Hello, okay. Welcome to day one officially of revision. So far in terms of year 13 biology content, we've gone through photosynthesis, but we're gonna go through it like in class. And just last week we covered respiration, aerobic respiration. So I think what we're gonna do today, because we were doing some past paper questions, and a massive roadblock for me was ATP. So I think what we're going to do today is very quickly go over a couple ATP questions, and then we'll quickly go over respiration. I'm talking, glycolysis, link reaction, Krebs cycle, oxidative phosphorylation. So, the structure of ATP is very similar to DNA. In fact, that was a very devious question that I set on this week's poll or quiz all about it. So, those are our phosphorus ions or phosphates. I actually don't recall what the structure looks like in terms of I think it's something. Honestly, I forget how the structure looks like, but this is all of the structural components, okay? Just, it probably looks wrong. So you have your adenine base, obviously it's adenosine triphosphate, those are your triphosphates, and that is a pentose sugar. I don't recall specifically which one it is, but I think in an exam you could probably get away with just saying it's pentose. So, let's break down the properties of ATP, shall we? There are a lot of properties, but there's only a few that they'd really want you to waffle about in an exam. Let's talk about, first of all, the phosphate ion, or the whole idea of phosphorylation. Phosphorylation is basically the addition of an inorganic phosphate, or PI, to make substances more reactive. So that right there, if you get asked, what is the importance of ATP in humans or something like that, that could be a good point to add. Or something about why is ATP needed. Like, I don't know what the context of a question would be, but any question where it's asking you to describe the purpose of ATP, it isn't just an energy compound, it also is used for phosphorylation. Next, the whole idea is that when this phosphorus, or I think it's a covalent bond, is broken, when this covalent bond is broken, it releases energy. But a big thing that people get wrong is that they assume that it releases all of the energy at once. It doesn't. It only releases small amounts of energy at a time in a simple reaction. And that right there, if I can actually find a pen, there we go. This right here is 2 points. So it's small. That's 1 point. Small amount of energy is released at a time. It doesn't just release it all at once. Another thing about ATP is, as I just said, it's a simple reaction. As all it involves is one bond being broken. The whole idea with that too means that it can easily be reformed. All it needs is ADP and a PI, so it can very easily be reformed in a variety of ways. So, those are 3 properties of ATP. Let's think about some more general ones. Firstly, the whole idea that this is a soluble molecule. It's a soluble molecule. Another one is the idea that it can't be stored. Now, the whole idea with it not being able to be stored is that is why humans need to generate so much ATP. Pretty sure I read humans need to generate more than their body mass of ATP every single day. And the reason for that is because it's not like one day we can just make a shit ton of ATP and then the next day we make none and just live off reserves. That's not how it works. ATP can't be stored. I'm pretty sure it also can't travel from cell to cell. So it has to kind of be used immediately. It's an immediate source of energy. So that is ATP. So that is everything you need to know about ATP, I'm pretty sure. Very quickly though, we could also talk about its connection to DNA, if you wanted. In DNA, it's deoxyribose nucleic acid. Deoxyribose instead of your ribose sugar. That's it, it's ribose. So deoxyribose is in DNA. Ribose is here. Adenine is here. In DNA, it just has any nitrogenous base. So it could be adenine, it could be thymine, it could be cytosine, could be guanine. Uracil is an RNA exclusive. And in DNA, there's only one... Actually, is there? Yeah, there's one phosphate. Because the base would be there in DNA. I remember. I'm good at remembering. Okay, so that is everything to do with ATP. I'm going to move away from the board so that we can look at all those properties. So, I think we should quickly do 2 or 3 exam questions. See if there's anything that the mark schemes tell us that we need to pick up on. Because you guys might not do what I do in terms of revision. You might not get my revision style, but I like it. It's this idea of dumping the information that I already know onto the board, and then by doing exam questions, adding information. So I'm going to go to Physics Math Tutor, which is a godsend for revision. I can promise you guys, if you're struggling with biology revision, a specific subject, please just Physics Math Tutor, you know. Right. It's going to be under biological molecules, which is a paper 1 topic. I think it's a paper 2 topic too. Or at least ATP can appear in various forms. Okay. So, let's answer some of these questions, shall we? Describe how an ATP molecule is formed from its component molecules. Interesting. That's about the ATP cycle, which I actually have not written down here. But let's see if we know what it is. Pretty sure it's just ATP, then it's ADP plus PI, and it's ATP again. I think that's it. And obviously this releases a high amount of energy by the breaking of covalent bonds. This requires ATP synthase. So, let's see if we can get 4 marks out of this. 4 marks for this, I feel like that's quite a tough ask, to be honest. Don't think we can get 4 marks, but let's give it a go. So, I'm going to say, should we say the PI in ATP, would it be hydrolyzed? Let's just say broken. Oh, okay, bond. We need to talk about bonds here, don't we? So let's say the covalent Honestly, I'm not sure about this. So let's look at the mark scheme. Okay, number one. I misread the question. Of course, it wouldn't be biology revision if I didn't misread a question. Describe how an ATP molecule is formed from its component molecules. I just talked about PI, ribose and adenine. I didn't talk about that. So I would have gotten 2 marks for literally just saying ribose and adenine. No! I fucked this question so bad. Oh my god, I fucked this question so badly. Oh my god, I overthought that question so bad. Holy shit. Wow. Okay, if the question had been, describe how an ATP molecule is formed, or if the question had linked in some way to the ATP cycle, I probably would have gotten 4 marks. I got no marks there, 0 out of 4. That is nasty. What I should have put is the 3 components. It's ADP and PI is via a condensation into ATP. Oh, via ATP synthase. Okay, so I would have got no marks there, but that is fine because I misread the question. It happens all the time, especially in biology, because they are so pretentious with the mark schemes. I've had dozens of streams, guys, where I've thought that I've nailed a question, looked at the mark scheme and gone, how the hell did they get to that conclusion? It's because you misread the question. You don't focus on a specific keyword, or you jump to a conclusion before you've taken time to think. So let's see if we can... Okay, here. Water is used to hydrolyze ATP. Name the 2 products of ATP hydrolysis. Okay, hold on. I don't want to overthink this, but hold on. Hydrolysis is the addition of a water molecule to split molecules. Condensation is the removal? Okay, I'll go with ADP and PI, but let's see whether it was water molecule. Oh, ADP and PI. Okay, cool. Okay, so I wasn't overthinking there. Cool. Hydrolysis of ATP is catalyzed by the enzyme ATP hydrolase. We know that. A student investigated the effect of ATP concentration on the activity of ATP hydrolase. Right, let's very quickly draw a graph. As you increase the concentration of a substrate, eventually the graph plateaus out. So just keeping that in mind. She took 4 slides. Oh, hold on. She used shortening. She used shortening of strips of muscle tissue caused by contraction as evidence that ATP was being hydrolyzed. She took 4 slides, A, B, C, and D, and added strips of muscle tissue to the same length of each slide. She then added the same volume of ATP solutions of different concentrations to the 4 slides. She recorded the final length. So hold on. So if the muscle tissue gets shorter, that's evidence that it's been hydrolyzed? I don't understand. Other than those given, give 2 variables she should have controlled. Okay, should we be obvious here or should we not be? I'm going to say temperature. Like room temperature, I'll say. I don't know whether that would be counted. And shall we say... I guess species that the muscle tissue is obtained from, or like the source of the muscle. I don't know how you would word it. Let's see. Species of muscle, species/organism a muscle came from. Temperature of the muscle tissue. Okay, so you would need to specify. So I would have gotten a mark. Temperature of the muscle tissue. Okay, that's an easy fix. So whenever you're stating a variable, what's it of? pH of soil, temperature of muscle tissue. You can't just say temperature because, as teachers love to point out, you could be waffling about anything. I get it. Describe and explain the pattern shown by the data for 2 marks. I forget how you do these questions. Usually it's like, this shows this because this. So hold on. So, do you need to give results if it's saying describe and explain? Description. The higher the concentration of ATP solution, the smaller the mass of muscle tissue. Sorry, the smaller the length. I don't know why. Why? As concentration of ATP increases, length of muscle decreases. That would be a mark. More ATP, so more energy released, so more muscle contraction. I get it, okay. Okay, that makes sense. Obviously, I could have probably put more ATP means more energy being released, because it's a hydrolytic enzyme. But no, I wouldn't have understood the correlation there between muscle tissue contracting and energy being used. But okay, that makes sense. Because when you're relaxing, it uses no... Of course, when you're contracting, it uses no energy. Sorry, when you're relaxing, it uses no energy. When you're contracting, it does. Okay, I get it. I understand that. That's a calculation, which I can't do. Scientists investigated treatment of a human bladder infection caused by a species of bacterium. The species is often resistant to antibiotics currently used for treatment. They investigated the use of a new antibiotic to treat the bladder infection. The new antibiotic inhibits the bacterial ATP synthase, cool, which represents the reaction catalyzed by ATP synthase. So is it the addition of a water molecule? That would be then ADP plus PI is ATP plus H2O, no? Yeah. Wait, what? Yeah, sorry, I misread the answers. I was right, my bad. I was confused for a second. Okay, the new antibiotic is safe to use in humans because it does not inhibit the ATP synthase found in human cells. Suggest why ATP synthase is not inhibited. Okay, so this is going to be because the antibiotic has a specific complementary shape which only binds to the ATP synthase in bacteria. So the bacteria has a different shape. I don't know how you would word it, but yeah. Human ATP... Oh, okay, you could talk about the tertiary structure. And active site. Why didn't I mention that? It's a protein. It has an active site. Yeah, it's a fuck. ATP synthase is an enzyme and a channel protein. I always think of it as being a channel protein. It's an enzyme too. It has an active site. Fuck. Human ATP synthase has a different tertiary structure. Has a different shape. It's not... Okay, antibiotic, not complementary. So I could have gotten a mark. ATP is an energy source used in many cell processes. Give 2 ways in which ATP is a suitable source of energy for cells to use. Okay, number 1, the energy... It releases energy in small amounts. I don't know why that would make it useful, but not all the energy is released at once, so it's released in small amounts. Number 2 is that... Why is it a suitable energy source? Could you literally put that it's soluble? Or would you have to say something like it can easily be reformed? Let's look at the mark scheme. Okay, no, sorry, so I overthought the question there. So yeah, it's right. Releases relatively small amount of energy. Little energy lost as heat. Interesting. I didn't think of that. So, should we look at... Okay. This is suitable energy. That's a reason why it's a suitable energy. That is a reason why it's a simple energy. Simple energy. Can be rapidly resynthesized. Even that does not leave cells. Does not leave cells. That is a reason why it is a good source of energy. And finally, the fact that it can be... It says rapidly resynthesized. So instead of that, let's say... rapidly resynthesized, and finally, is not lost. Cells constantly hydrolyze ATP. Describe how ATP is resynthesized. So that would be the condensation reaction between an ADP and a PI catalyzed by ATP synthase. Give me my marks. Yep, from ADP and PI by ATP synthase. Fuck, during respiration or photosynthesis. You couldn't say condensation? That's a stupid mark. Give 2 ways in which the hydrolysis of ATP is used in cells. Okay. The energy released from the hydrolysis of ATP can be used in various metabolic reactions. Phosphorylation is the addition of an inorganic phosphate which makes substances more reactive. Give me my marks. To provide energy for other reactions, to add phosphate to other substances to make them more reactive. So you might not have gotten a mark for what I said there. I said provide energy for metabolic reactions. I guess just other reactions. Cool. So, that's all I'm going to do today, to be completely honest, because I am going to do some more questions in my own time. I think we've done quite well there. We've done a lot. So, the structure of ATP, we know off the top of our heads. That's piss easy. We know that it's soluble. We know that it's an immediate source of energy. We know the whole thing about phosphorylation. What we didn't know before this, really, or what we forgot, sorry, is it's a condensation reaction. We know now that it's a simple reaction, which can be rapidly resynthesized. We know that only small amounts of energy are released at a time. We know that it can't leave a cell. We know that it can't be stored. And I think that's about it. Peace.